So good afternoon to everyone, and thank you very much, Arian, for this kind introduction. So we know that tumor protein 53 expressed from TP53 gene on chromosome 17 is one of the most important tumor suppressors. And the frequency of TP53 mutation in myeloid neoplasms is up to 10% for MDS and AML, and even higher in those patients affected with therapy-related diseases. And across all these clinical scenarios, we know that the presence of TP53 mutation means very aggressive disease, high risk of disease evolution, poor outcome, and refractoriness to conventionally available treatment. So in the first part of this presentation, I would like to share with you some key and conventional concepts in order to interpret in the right way the biological and clinical effect of TP53 mutations in the clinical scenario of myeloid neoplasms. And as the first point, we and others clearly observed that whether TP53 mutations are clonal or subclonal does not influence the effect on patient prognosis. So in other words, also small TP53 clone matter in terms of driving a poor outcome of patients with myeloid diseases. The second clinical concept, firstly observed by Elsa Bernard a few years ago, is that TP53 allelic status is associated with a clear prognostic relevance, especially in the context of chronic myeloid neoplasms such as MDS. And as you can see here, according to the results of this study, only patients with biallelic TP53 inactivation are associated with a very poor outcome, while patients with monoallelic TP53 inactivation present a similar outcome as observed in wild-type individuals. According to this strong evidence, both ICC and WHO current classification systems agree to define distinct clinical entities within MDS and AML clinical scenarios associated with the presence of a biallelic TP53 inactivation, despite some discrepancies in the criteria that are suggested in order to correctly identify this patient in a routine clinical practice. So in the second part of this presentation, I would like to move to discuss with you new insights in order to possibly refine the biological role of P53 mutation and dysfunction within myeloid neoplasms as a basis for an optimized clinical management of the patient. And the first contribution comes from Luca Lanino and Elisabetta Sauta from Yale University and Humanitas Research Hospital, respectively, which are coordinating the TITAN study. It is a collaborative worldwide effort aimed to collect and analyze the clinical and genomic information from very large real-world-based cohort of patients affected with myeloid neoplasms. And as you can see here, we collected more than 20,000 patients with complete clinical and genomic information available, together with matched RNA sequencing information from bone marrow progenitors in a significant proportion of the patient for correlative analysis. And with respect to the context of TP53 mutations, we clearly demonstrated that there is a cluster of patients associated with the presence of biallelic TP53 inactivation, in which the assignment of the patient to this cluster is irrespective of diagnostic entity and blast count. As you can see here, AML, MDS, and CMML patients contributed to the same cluster. The prognosis of the patient is really poor and is comparable in a different patient with a different clinical phenotype. And even more importantly, the gene expression profile of this cluster is really consistent with a strong dysregulation of P53 pathway in all the different clinical entities. So overall, this data might suggest the possibility to define a unique clinical entity across all the myeloid neoplasms defined on the basis of a specific genomic abnormality and without any additional contribution from conventional clinical and histological features. The second contribution was provided by Matteo Zampini from our institution, who was able to refine a little bit the right definition of the prognostic relevance of TP53 allelic status in MDS. Matteo analyzed a large cohort of more than 7,000 MDS patients and was able to demonstrate that the prognosis of patients with monoallelic TP53 inactivation is intermediate as compared to wild-type patients and patients with biallelic inactivation. And this is also true, as you can see here, in patients receiving disease-modifying treatments such as hypomethylating agents and allogeneic stem cell transplantation. Importantly, the prognostic effect of mono and biallelic TP53 status was confirmed in a multivariable analysis and was irrespective from variant allele frequency of the gene. So there is growing evidence that the definition of TP53 status is a driver for specific clinical decisions in this peculiar population of patients. We know, for instance, that for those patients that are eligible for a transplant procedure, the presence of TP53 mutation is a potent driver for a high risk of disease relapse after a transplant procedure, especially for those patients carrying biallelic gene inactivation. But despite that, recently, in a trial that was associated with a biological randomization of the patient in performing allogeneic stem cell transplantation on the basis of the availability of a suitable donor, TP53 mutated patients treated with allogeneic stem cell transplantation are associated with a longer life expectancy as compared to those patients receiving non-curative therapeutical approaches. In the same context of allogeneic stem cell transplantation, we did another potentially important observation. And in our institution, recently, Cristina Tentori developed and validated an innovative decision support system based on artificial intelligence, including both clinical and genomic information to define the optimal timing of transplantation for MDS patient population. And with respect to the patient with TP53 mutation, we clearly observed across all the patient ages categories that performing transplantation within a monoallelic TP53 inactivation status was associated with a longer life expectancy as compared to transplantation performed in patients with TP53 biallelic inactivation. So this data may provide a sort of relevant information in defining the optimal timing to perform a transplant procedure within this patient population. And also stress the point that mutational landscape of TP53 is potentially dynamic during the natural history of the disease. Why it is important to talk about optimal timing of transplantation in myeloid neoplasms? And these questions was addressed by Luca Lanino, who recently analyzed a large cohort of more than 800 patients with CMML receiving allogeneic stem cell transplantation. And Luca applied the decision support system developed by Cristina in the previous slide in order to define which patients were transplanted within the optimal timing as defined by the decision support system, as compared to those patients in which transplantation was performed outside the optimal window. And as you can see here, performing transplantation within the optimal timing window was associated with a gain of overall survival of more than 2 years. And this relevant effect of the optimal timing on the post-transplantation outcome of the patient was maintained in a multivariable model adjusted for all the patient and transplanted related features. So now the next question that is relevant in order to define the clinical and biological role of TP53 mutation within myeloid neoplasm is whether monoallelic and biallelic status are in some way static stages, or there is a dynamic possibility to shift from one state to another during the natural history of the disease. And in order to address these very important questions, Matteo Zampini provided a study on paired bone marrow samples from a small series, 42 patients of MDS with the TP53 mutation collected both at the time of diagnosis as well as at the time of leukemic evolution. And as you can see here, in 23 patients with monoallelic TP53 inactivation at the time of diagnosis, in most of the cases, the progression to AML evolution was associated with the progression to a biallelic status of TP53 inactivation, thus suggesting that finally, mono and biallelic inactivation are different stages possibly occurring during the natural history of the disease. So this means that TP53 mutations are associated with clonal evolution during the natural history of the disease, and we know more in general that all MDS and all chronic myeloid neoplasms are exposed to the risk of clonal evolution during time. With respect to this situation, we have to say that currently available prognostic systems are associated with a major limitation, since they account for clinical and molecular information collected only at the time of diagnosis, thus missing the opportunity to include longitudinal information associated with the possibility to capture the clonal evolution of the disease. In order to possibly address this issue, recently, Ivan Civettini in Milano provided a huge effort in order to build, as a proof of concept, a new prognostic score for MDS that is based on molecular evolution, and that was called IPSS-M-EVO. Ivan developed a specific algorithm that is able to do 3 different things. As a first point, the algorithm is able to estimate the cancer cell fraction for each mutation and to assign a temporal order among the driver mutation during the MDS evolution. As a second point, the algorithm is able to identify evolutionary trajectories within an MDS population, and last but not least, these trajectories were integrated into the conventional molecular IPSS, resulting in the IPSS-M-EVO score. This new score was trained in a large population of patients from available public data set and also validated in an external cohort of MDS. Importantly, we identified a lot of evolutionary trajectory, including a specific trajectory focused on TP53 dynamic evolution. And finally, the model with EVO was associated with a significant improvement in the definition of the individual risk of both leukemia-free and overall survival, with more than 40% of the patients that were restratified according to the conventional prognostic assessment. So overall, as you can see here, the new score based on molecular evolution is able to outperform conventional molecular IPSS across all the clinically relevant endpoints, thus suggesting that for the next future, the next generation prognostic score for myeloid neoplasm should be based on the evaluation of longitudinal data. This is maybe the most important information that I would like to share with you during my presentation. And to me, the most important clinical question is whether the prognostic role of mono and biallelic inactivation for TP53 is limited to the scenario of myeloid neoplasms, or as an alternative hypothesis, we are facing with a more general phenomenon that is also able to stratify a large number of additional hematological diseases. And in order to answer to these very important clinical questions, Luca Lanino at Yale University collected a huge number of clinical and biological information from publicly available repositories across different hematological malignancies, including not only myeloid disorders, but also lymphoproliferative diseases. And as you can see here, the prognostic stratification of the patient based on the definition of the allelic status of TP53 was maintained also in chronic lymphocytic leukemia, in diffuse large B-cell lymphoma, and many other hematological diseases. So just suggesting that this dynamic evolution across different stages of allelic inactivation of TP53 is maybe a general phenomenon associated with a strong biological and clinical effect across potentially all the myeloid malignancies. In the very last part of the presentation, I would like to share with you the concept that there is an emerging evidence that P53 dysfunction in myeloid neoplasms can be associated with alternative mechanisms with respect to the conventional mutation of the genes. And again, Matteo Zampini in our institution, by using RNA sequencing approaches, was able to identify within a large cohort of MDS patients a population of individuals accounting for around 5% of the whole MDS population that was associated with a clear dysfunction of P53 pathway, but without any evidence of TP53 mutation, even performing a deep and ultra-deep gene sequencing. And for sure, in this patient, there is a strong dysregulation of the downstream P53 pathways that is similar to that observed in patients with biallelic conventional TP53 inactivation. And as you can see here, we provided a proof of concept of alternative mechanisms in terms of dysregulated biological pathways within the cells that may be responsible for this non-mutational dysregulation of P53. What is important in terms of potentially recognizing this patient in a clinical practice is that we observed by immunohistochemistry, by using P53 staining in the bone marrow, that this patient can be easily recognized by an abnormal expression of P53 in hematopoietic progenitors. Why it is important to account for these additional non-mutational mechanisms associated with P53 dysfunction? The reason is that, as you can see here, in MDS, for instance, the prognosis of this non-mutational dysfunction patient is comparable to that of patient with biallelic inactivation. And this is also true outside the setting of MDS, for instance, analyzing public repositories of patient with AML. As a last point, in collaboration with Sharon Cordasti at King's College, we provided an additional contribution to the general concept that TP53 mutated myeloid neoplasms are associated with a distinct immunosuppressive profile that is one of the most relevant drivers for the poor prognosis that is observed in this patient and may represent a source of information to develop innovative treatments and to check for specific vulnerabilities within this challenging population. And together with Sharon, we analyzed by multi-omics single-cell approach paired samples in a population of patients in which bone marrow aspiration was available both at the time of diagnosis as well as at the time of leukemic evolution. And we observed a specific inflammatory signature in myeloid cells and T cells, most including Tregs, that is consistent with a distinct immune dysregulation involving myeloid-derived inflammation and impaired antigen presentation. Specifically, the downregulation of BATH2 and the subsequent enrichment of CD161 positive Tregs with very high suppressive capacity really suggests that chronic myeloid-derived inflammation may drive an immunosuppressive state in this patient with the TP53 mutation that is really independent of interferon gamma pathway. And I think that it is important to continue in the biological and immunological studies of the specific dysregulation in this population as a source for potential information for development of innovative treatment. So to summarize, in myeloid neoplasms, TP53 mutations are associated with poor outcome and resistance to currently available treatment. We discussed that both clonal and subclonal mutations are associated with a negative prognostic effect. TP53 mutations typically occur as a multi-hit process, and we provided preliminary evidence that mono and biallelic inactivation are different stages occurring during the natural history of the disease. The TP53 allelic status has a prognostic significance that supersedes variant allele frequency and other clinical biological features. So the TP53 allelic status may inform the optimal timing of therapeutical intervention with a specific focus to allogeneic stem cell transplantation. And outside the clinical setting of myeloid neoplasms, TP53 allelic status is emerging as a powerful prognostic determinant across multiple hematological diseases, including CLL and lymphomas. So we discussed that the next generation prognostic systems for myeloid neoplasm should be based on longitudinal genomic data and should be based on disease evolution dynamic and trajectories as provided as proof of concept by the recently proposed IPSSM EVO. Last but not least, we discussed that non-mutational mechanisms may contribute to P53 dysfunction across different myeloid neoplasms, and that P53 dysfunction is associated with a severe immune dysregulation, suggesting a complex interaction between genomic instability, inflammation, and immune surveillance. What are the next steps? I would like to highlight two different initiatives in order to further improve our understanding of the biological role of P53 in myeloid neoplasm. The first one is provided by I4MDS consortium supported by EHA. We defined a specific team totally devoted to TP53 in order to improve the understanding of the disease biology. And recently we also launched the so-called Nobel Prize Initiative in MDS, originally provided by Humanitas, MD Anderson, and King's College. And of course, this initiative is open for additional partners and collaboration. And the main objective of this initiative is to define within the European healthcare data space a specific repository for multi-omics information in MDS with a specific focus on TP53 to provide the proof of concept for new disease vulnerabilities as a basis for innovative treatment. And thank you very much for your attention.
Thank you, Mattea. And your excellent talk is the start of a Nobel Prize winner, maybe. That would be great. So you showed a lot of new data, also data just published yesterday. You can read it in GCO. But the floor is now open for discussion. Who may start? The room is full, and also online we have over 160 participants. So who will start? Maybe I can start, Matteo.
Yes.
You mentioned several times the optimal timing of transplant in the TP53 setting. What is optimal?
Yes, to me, just to provide a simple concept, is that in a patient with monoallelic TP53 inactivation, we don't wait for an evolution to biallelic status, and maybe anticipating the indication to perform transplantation can be associated with a better post-transplantation outcome.
Okay, but then you should also argue the time, and I missed it in your slide, from monoallelic to biallelic. What is the median time of progression in that setting?
Yes, this is a very important question. We have no data in order to define what is the exact individual trajectory in terms of the risk of evolution into the biallelic status. But I think that the proof of concept of this kind of longitudinally based next generation prognostic systems can provide us for the opportunity to capture the individual risk of evolution in terms of clonal progression of TP53 allelic status as a basis for a more appropriate clinical management of the patients.
Thank you. Microphone 2 in the front here.
Me?
Yeah,
all
please.
Right. I thought about excellent talk. Very intriguing, and I think it's a very important question to ask. So can I ask, you mentioned about those non-mutational TP53 dysfunction. So if there's no mutation, how do you discover those cases? There's a functional assay, or I saw some immunohistochemistry on the right-hand side. So how do you discover those cases?
Yes, so the first observation in order to provide a concept for a non-mutational dysfunction of TP53 was provided by AI clustering, because we identify a cluster of the patient with potentially a biological similarity in which both TP53 mutated and non-mutated patients were together in the same group of patients. And we check for potential similarities between patients with or without mutation that were classified in this cluster. And we found that one similarity was the hyperexpression of P53 by immunohistochemical staining in the bone marrow. And by RNA sequencing analysis, we found that also in non-mutated patients, there was a strong dysregulation of P53 pathway. This was the rationale in order to study specifically this patient population and to define the proof of concept for non-mutational P53 dysfunction that is really already observed in other human cancers. This is not a novelty, but just to say that what is true for many solid tumors in terms of defining non-mutational perturbation of P53 pathway, it is also true in a small fraction of patients with myeloid neoplasms.
So am I correct in a nutshell?
Sorry, may I interrupt you we have only 1 minute left. So on the back, right, please, go ahead.
Hi, thank you very much for the talk. I have a question regarding germline TP53 mutation. Do you regularly test for it? And what is the role of germline TP53 in the IPSSM?
Yes, so this is again a very important question. So it is really established that TP53 can be a germline mutation as well as a somatic mutation. I think that both mutations are relevant in order to define patients with high risk of disease evolution and poor prognosis. Currently, to my knowledge, the information about germline mutation of TP53 is not yet included in the prognostic assessment of MDS or other chronic myeloid neoplasm, but it is a good point. We are moving to a more comprehensive genomic screening in MDS, not only including somatic mutation, but also germline genomic events are so relevant in driving the disease evolution, the prognosis of the patient.
Okay, very short question and very, very short answer.
Okay, very quick. Very struck by the data that the clinical outcome between the mono and a biallelic with azacitidine is not as hugely different as you saw in the allogeneic stem cell transplant. So did you observe there was difference in the immune inactivation in the setting of a biallelic P53 mutation, which is what is driving the less GBL when it's biallelic versus monoallelic?
Yes, I think that, again, we have not yet sufficient data in order to define a clinical recommendation, but my message is that maybe it's not totally true that monoallelic inactivation status is not associated with any particular relevance in terms of different prognosis of the patient. Maybe we are talking about an intermediate state, so we need to collect more data, especially longitudinal information, in order to check the proper disease stratification according to the allelic status.
Okay. With respect to the EHA members and speakers, I have to stop now. Thank you, Matteo. And we move on to David Salman. David, take the